Hello everyone, in this video we'll see about metagenomics in detail. Metagenomics is the study about genetic material extracted directly from environmental samples like soil, water or the human gut. But in traditional microbiology, the samples are cultured and grown in labs and it is studied. But metagenomics involves the study of microorganisms that cannot be cultured or produced in lab. The benefits of metagenomics are it helps to discover microbial diversity and it helps to identify novel genes and their metabolic pathways, and it also helps to understand the role of microbes in health, environment and industry and their interactions. Now let us see the actual definition of metagenomics. Metagenomics is defined as the genomic analysis of microorganism by direct extraction and cloning of DNA from an assemblage of microorganism. From a mixed microbial population, this metagenomics analyzes the collective genomes and provides information about their structure, function, and their interaction. Now, let us see the history of metagenomics. In 1991, the idea of analyzing mixed microbial DNA was introduced. Later, in 1998, the word metagenomics was first used by Joe Handelsman and his colleagues. In 2000, the rise of next generation sequencing method gave a very big transformation in the metagenomic studies. Now we'll see the need of metagenomics. The main need of metagenomics is we can access the hidden microbial diversity in various environments, and also we can study the functions or characteristics of many microbial communities when they are present in their natural habitats itself. And it also used to discover many new genes and enzymes with industrial and pharmaceutical potential. Metagenomics also helps us to understand microbial roles in nutrient cycling, health and disease. The next topic is steps in metagenomic analysis. The steps involved are sample collection, DNA extraction, sequencing, bioinformatics analysis, and data interpretation. In sample collection, the sample which we want to analyze is collected. Samples include soil, marine water, gut, or sediments. Next, the DNA is extracted from the sample. The extraction process is either chemical or mechanical methods. Now, the extracted DNA is sequenced. High-throughput screening methods like Illumina or nanopore methods are used for sequencing the extracted DNA. The next step is bioinformatics analysis. Here, computational tools are applied to assemble, annotate, and compare the sequence that is sequenced using high-throughput screening in the previous step. Later, the results obtained by the bioinformatic analysis is interpreted and the microbial diversity, taxonomy and other functions are identified. Now, let us see the techniques used in metagenomics. There are three different techniques used. One is DNA extraction, next is sequencing technologies, and last is bioinformatics tool. Techniques used in DNA extraction are bead beating, enzymatic lysis, and phenol chloroform methods. So, these methods are used for extracting the DNA from the sample obtained. Mostly, the commercial kits are always used for purity and yield. In sequencing technologies, Sanger sequencing was used in early days, but it had some disadvantages. But today, next generation sequencing methods like Illumina or Oxford Nanopore is used. These methods provide easy and quick results. Different bioinformatic tools are used based on the parameters that we want to study. Applications of metagenomics. The first one is human health. Using metagenomics, the human gut microbiome and its role in digestion, immunity and disease is studied. Also, the microbes that are linked to obesity, diabetes and mental health is also studied. Metagenomics is also used in the agriculture field. Here, the soil microbiota is studied for improving the crop yield. We can also identify the nitrogen fixing and plant growth promoting bacteria. In environmental science, the study of metagenomics is applied in the bioremediation process. Here, the microbes that degrade pollutants or plastics is studied. Also, we can study the roles of microbes in carbon and nitrogen cycle. Now, we'll see a case study to understand the metagenomic process in detail. A project called Human Microbiome Project was started, and they analyzed thousands of microbial species in the human gut. As the result, they found that the human gut contains over 3 million microbial genes, far more than the human genes. Using this project, they came to know about the relationship present between the gut microbiota and the disease like obesity, diabetes and depression. So, the Human Microbiome Project helped in the development of many probiotics and personalized nutrition or personalized medicines. Thank you all.